Now we'll see about the endodontic microbiology. If you see the portals of entry for microorganisms, that is, entry through the open cavity. This is the most common way of entry of microorganisms into the dental pulp. So when the protective layers of the tooth such as the enamel and the dentin get destroyed by the caries or by any injury, the microorganisms gain access into the pulp cavity. Next we'll see the dentinal tubules. Here the microorganisms will pass through these dentinal tubules subsequently to the pulp. So what happens here is that the microorganism's diameter is about less than 1 micrometer, where diameter is that 1 to 4 micrometers. So that's how the microorganisms can easily penetrate into these dentinal tubules. Okay, next we'll see the other opening is through the periodontal ligament or through the gingival sulcus. During the periodontal pathology or the treatment which removes the cement and exposes the dental tubule to the oral flora through which the microorganisms gain entry into the pulp through as from the lateral canals. Now we'll see what is anachorasis. It is nothing but the process by which the microorganisms are transported in the blood to an area of inflammation where they establish an infection. Next is through the faulty restorations. The faulty restorations with the marginal leakage can result in contamination of the pulp by the bacteria. If you see, the different type of microorganisms available are the cocci, in which we have the coccus and the staphylococcus and streptococci. And the bacilli, in that we have variety of bacilli such as the Bacillus, Diplobacillus, Palisade and the Streptobacilli. And others such as we have the Corynebacterium, Vibrio, Corynea and the filamentous bacteria. Next, the microbial classification. The classification of the microorganisms, we have variety of microorganisms in that the obligate anaerobe, and it includes that the organisms requiring the oxygen for their growth and they cannot ferment or respire anaerobically. Example are the tubercle bacilli. And the faculty anaerobes are the organisms which grow in the presence of the or the absence of the oxygen. Example are the staphylococcus. And the microaerophilic are the organisms which grow in the oxygen environment but derive their energy only from fermentable pathways that occur even in the absence of oxygen. Example are the streptococcus. The obligate anaerobes are the bacteria which can grow only in the absence of the oxygen. Example are the bacteroids and the fusobacterium. And the microbial flora are classified on the basis of the gram-positive or the gram-negative organisms. That's how the classification of the microorganisms occurs. If you see the microbial virulence and the pathogenicity is nothing but the ability of the microorganisms to produce a disease, whereas the virulence is a degree of pathogenicity of the microorganisms. So, Hobson gave an equation showing the relationship of the number of microorganisms, their virulence, and the resistance of a host with the severity of the disease. That includes the number of microorganisms is multiplied by the virulence of the organisms and divided by the rest of the host, which gives us the severity of the disease. If you see the virulent factors, in that we have the lipopolysaccharide which is present on the surface of the gram-negative bacteria, which cannot be neutralized by the antibodies and they exhibit the biological functions in the form of the endotoxins, which have the capability to diffuse into it. Next is the extracellular vesicles. They are produced by the gram-negative bacteria in the form of endotoxins, which releases into the dentin. And the enzymes produced by the bacteria can result in the spread of infections, neutralization of immunoglobulins, and complement components. And the fatty acids are like propionic acid and the butyric acid are produced by the anaerobic bacteria, which helps in causing the neutrophil chemotaxis, which results to the perirrhizis. And the polyamines are the biologically active chemicals found in the affected canals. And other virulents such as the villi and capsules are also present in the gram-negative. If you see the factors influencing the growth and the colonization of the microorganisms, it is based on the influence of oxygen, that is the low availability of the oxygen in the infected microbial root canals, which causes the high facultative organisms in the root canals. And the nutritional factors that play an important role in the bacterial colonization, which is the source of nutrition for the bacteria. And the bacteriocins. Some bacteria produces the bacteriocins, which are antibodies like proteins produced by one species of the bacteria to inhibit the another species of the bacteria. And the co-aggregation is the existence of the symbiotic relationship between the some bacteria, which increases the virulence by the organisms in that ecosystem. And the bacterial interrelationships are necessary between the certain bacteria, which is antagonistic, or the commensal, which affects their survival. And the microbial ecosystem of the root canal, if you see that the microorganisms can survive in the environment of the low oxygen tension, which can survive the limited pabulisms which are found in the root canals. And if you see here, we have the certain bacteria and its interrelationships, which are the key factor for the growth of the one organism, that is a one bacterial species to another bacterial species. And these all occur under the low oxygen tension from the necrotic tissues. These are the certain bacteria which we can see here. And if you go for the type of infections caused by these microorganisms, that is summarized as that by the NIDOF, that the endodontic flare-ups and as well as the asymptomatic cases are almost similar. And the pulp contain the flora almost similar to the oral cavity, and mixed infections are more common in than the single organisms. If you see the types of endodontic infections, we have the intraradicular as well as the extraradicular. The intraradicular infections are the infections in which the microorganisms are present in the root canal systems. The primary intraradicular infections are the infections which invade the pulp tissue, resulting in inflammation than the necrosis. And these probably are dominated by the gram-negative anaerobic bacteria such as the Porphyromonas, Provatella, Fusobacterium, Campylobacteria, and the Trypanoma. And the secondary infections that occur during or after the treatment. That may be during the course, that is the remnants of the caries or the contamination through the instrumentation. These includes the bacteria such as the Pseudomonas aeruginosa, Staphylococcus, Escherichia coli, Enterococcus faecalis, and the Candida species. And the persistent intraradicular infections are the infections in which those microorganisms can survive in the root canal after the intracanal antimicrobial procedures. In these infections, the E. faecalis is most commonly found organisms. The E. faecalis is considered to be the most common reason for the failed root canals, and even in the canals with persistent infections. And due to its features such as that it can persist in the poor nutrient environment of the root canal treated teeth, and it can survive in the presence of the medicaments like calcium hydroxide, and will stay alive in the presence of the irrigants like sodium hypochlorite, and it can convert into the viable but not non-cultivable state. So it can form the biofilms in the medicated fluids, and it can penetrate and utilize the fluid which is present in the dentinal tubules and survive the prolonged periods of the starvation and utilize the fluid that flow from the periodontal ligament. And it also survives the low pH with a high temperature, and also the extraradicular infections. It is characterized by microbial invasion and proliferation in the inflamed periradicular tissues as a result of intraradicular infections. Example would be an acute alveolar abscess. So, if you see, these extraradicular infections may be caused by an intraradicular bacteria or the bacteria from the outer side. So, common microorganisms in the extraradicular infections are Actinomyces treponema, P. gingivalis, Fusobacterium nucleatum, and Prevotella endodontics. That is Prevotella endodontalis. And if you see the identification of the bacteria, the test can be done to detect the microorganisms and their sensitivity to a particular antibiotic. And the Gram stain, which was developed by Christian Gram in 1884, helps in differentiating the bacteria in gram-positive and the gram-negative organisms. Next, we'll see is the culture. This is a method, though done during these days very less, but its importance is very much because of its wide range of bacteria found in the endodontic infections. So if you consider the principles of the culturing, there are two main reasons. In case of the persistence infections, to grow and isolate the microbial flora for antibiotic sensitivity and the resistance profiles, and to assess the bacteriological status of the root canal system before the obturation occurs. Okay, and if you see, there are various methods of culture technique. So the samples can be obtained either from the infected root canal or from the periradicular abscess. If you see the sample from the draining root canal, if there is a drainage, collect the sample using the sterile needle and the syringe or the sterile absorbent points. Place the aspirate in anaerobic transport media. And if you see the sample from the dry canal, here also use a sterile needle or place transport media into the canal. That's how we aspirate it into an anaerobic media. And for the case of the abscess, you have to palpate the fluctuant abscess and determine the most dependent part of the swelling and disinfect that area and penetrate a sterile 16 to 20 gauge needle in the surface and aspirate the exudates. Then transfer it to the anaerobic transport media. Now we'll see about the culture reversal. Sometimes the negative culture becomes positive after 24 to 48 hours, so it is advised to allow more than 48 hours between taking the culture and the obturation. And if you see, in the recent times, we have gone for the molecular diagnostic methods because this culturing method is sometimes gives us the false negative and also low sensitivity and specificity. So to overcome this, the molecular diagnostic methods have been came into the play because of its higher sensitivity and specificity, and else also helps in detecting the cultivable and uncultivable microbial species. And they are classified into four categories such as the hybridization, amplification, sequencing, enzyme digestion of the nucleic acid. If you see the first method, that is DNA hybridization method, in this method it uses the DNA probes which target the genomic DNA or the individual genes, which helps in determining the presence of multitude of bacterial species in single or multiple samples. So now we'll see the polymerase chain reaction method. In this method, it involves the in vitro multiplication or the replication of the DNA. Therefore, it is also called as genetic xeroxing method. As if you see in this, the multiple copies of the specific region of the DNA are made by repeated cycles of heating and cooling. And it also gives us highly specific and the cultivable and non-cultivable bacteria. So, now we'll see how to compare the microorganisms in the endodontic therapy. That is usually done by taking few measures, that is thorough cleaning and shaping of the root canal system and providing the oxygen to the canal by through the access opening. That's how these the bacteria which are anaerobic will get this oxygen and they cannot survive in this oxygen environment and they die. And use of the antibiotics with the intracanal medicaments which play an important role in combating the microorganisms, and also the use of calcium hydroxide in the canals with the necrotic pulps after instrumentation will provide us beneficial results. And later we can see the successful root canal therapy with a three-dimensional obturation with an healed periapical bone. So this is brief about the endodontic microbiology.